Good morning, students. Students, today we will see one more application of the biotechnology in the field of agriculture. And your application is flavor savor tomatoes. Now see children, what is the meaning of flavor savor tomatoes? Actually, we want, when we go to the market and buy tomatoes, then we want the tomatoes should be tight, okay, and it should be bright red in color. We never go for the soft tomatoes because if we'll buy them and if we'll keep them in our refrigerators, then within 1 or 2 days they all will become over ripened and destroyed. Okay, so what we want, we want that tomatoes should be of red color and it should be tight. Now, just to get tight and red tomatoes, the scientist, and even we can say that just to get tight, red and longer period of tomatoes, a periods of longer period which can remain tight and red for longer period. We want that tomatoes and we buy that one. Just to develop this type of tomatoes, one scientist came and the name was MacGregor. Okay, now what he did, he worked hard and on his experiments, by his experiments, we got tight red and longer period of time which can remain tight and red for the longer period of time. We got this type of tomatoes. Now children, see the basic behind this. Actually what happens, the tomatoes have the cell wall of these fruits, they have pectin. Okay, so what to write? The cell wall of tomatoes are made up of pectin. Okay, and to dissolve this pectin, there is one enzyme, and the name of the enzyme is polygalacturonase. Okay, so the outer surface of the tomatoes are made up of pectin and the outer surface has one enzyme named as polygalacturonase, also named as PG. And this PG is responsible for ripening of the fruit. So what is this responsible for? Ripening of the fruit. If PG is there, then what this PG will do? This PG will dissolve the pectin. So instead of ripening, what we can write? This PG will dissolve the pectin wall and responsible for ripening of fruit. Okay. So this is the general normal concept that what will happen? The cell wall of the tomatoes, they are made up of pectin and these pectin will be dissolved with the help of an enzyme named as PG. Now, this PG is, if this PG is responsible, then see what the scientists have done. I'm going to erase this. Now see what the scientists have done. The scientists have taken out the PG, okay, and go for its transcription. We know the process now that if enzymes are there, then what these enzymes will do, if they will help in the formation of protein, then definitely, like from the gene to the protein, transcription and translation will be there, formation of mRNA will be there. So what the scientists have done, they have taken out the gene of PG, okay, and then undergo for the process of transcription and formed mRNA, okay. Then with the help of reverse transcriptase, once again children, with the help of reverse transcriptase, they formed cDNA, complementary DNA, and this cDNA is of the gene PG, okay. So this is our one part that was done by the group of scientists. Now come to the normal process. What will happen in case of normal process in the tomatoes? The gene PG will undergo for normal transcription, okay, and form mRNA. After this, normal translation and the formation of protein, and the protein will be our enzyme, and the enzyme is once again PG only, okay. So now see what the scientists have done. They have done this step. They produced mRNA and they introduced this in tomatoes plant. As soon as the plant develops, this also developed, it forms cDNA inside the plant body, and now when the plant starts doing its normal process, then this mRNA was silenced by cDNA. Because it is of its same. It is same as the mylidogine incognita that we have already discussed. Okay, there also we have made the cDNA, introduced it into the body, into the tobacco plant, and as soon as the mylidogine infected, that cDNA was complementary. It was complementary. So this cDNA was silencing the mRNA. In the same way, children, this cDNA will silence the mRNA. And if mRNA will be silenced, it will not work, then there will be no formation of the enzyme PG. And if PG will not be formed, then definitely our tomatoes will remain red tight for the longer period of time. So this is the flavor saver tomatoes. These tomatoes are also called as the MacGregor's tomatoes because it was discovered by the MacGregor for the first time. Okay, so this is all about your application in case of biotech. One and last topic is left, children, and that is golden rice. Okay, now see what is the meaning of golden rice. We know people, in spite of having normal diet, they suffer from some of the deficiency diseases. Many of us, we are taking the nutrition, a diet which is full of nutrition, but still we see that some people are suffering from some of the deficiency diseases. Okay, so just to overcome that deficiency disease, what the scientists have started doing, they have introduced some nutritional values in the crops. Okay, and these type of crops are called as the bio-fortified crops. Like in case of wheat, we have made Atlas 66. Now here we are going to concentrate on the topic about the golden rice. Now see, children, what is the meaning of this golden rice? Okay, wait a minute. Yes, now see, out of the deficiencies, One such deficiency is deficiency of vitamin A. Okay, so we will see the deficiency of vitamin A. The deficiency of vitamin A can be overcome by using a technology called as golden rice. Okay, so by using golden rice, now the meaning of why we are calling it as a golden rice, because the rice looks like yellow in color. Okay, because of this yellow color, we gave a name of golden rice. Now see children, what is the deficiency of vitamin A and why we are taking this golden rice. We all know that beta carotene is a raw material for the synthesis of vitamin A. Okay, so I am going to write. We all know that beta carotene is precursor. Better we can call it as a precursor, that is raw material for synthesis of what? Vitamin A. Now concept must be clear to you all children. What the scientists must have done? They must have taken the gene of, okay? So they must have taken the gene of beta carotene synthesizing, like they must have taken beta carotene synthesizing gene. Clear? And introduced them into the rice. Now this beta carotene, let me write the full. This beta carotene is yellow in color. Okay, so what they did? They introduced the beta carotene synthesizing gene into the rice. Okay, and now rice has started producing rice with vitamin A. Okay, and that's why it looks like yellow in color and we call it as a golden rice. So this is the concept to make the crops or to fortify the crops, to increase the productivity and the nutritional value of the crop, so that we can get ourselves, like we can get the multiple nutritions. Okay, so this is the fact about the golden rice. Now children, along with this there are wheat, and that is we have developed one variety of the wheat, that is Atlas 66. This is the improved variety of the wheat. Along with this, people or the scientists are also working for iron fortified rice. Okay, and in this, once again, the content or the nutrition of iron is in the proper amount. Okay, in the new variety of the rice as the earlier one. So these are few more applications in the field of biotechnology. And now I think so application in the agriculture has been completed. Okay, so once again, if you will open your syllabus and if you will go through it, then you will be able to see that we have completed all the applications in case of agriculture. Yes, production of crop tolerant abiotic tissues or stresses, cold, drought and salt and heat, pest resistant BT crop, RNAi with reference to melagogyne crops, BT, and biofortification, golden rice, wheat Atlas 66, maize hybrid, and iron fortified rice. So in the same way, this is all about the applications of biotechnology in the field of agriculture. Clear, children? Now on this one. Beside maize hybrids that contain twice the amount of amino acids, lysine and tryptophan compared to existing maize hybrids were developed. Like this word I have not used, so I am telling you this also. The scientists have started producing the varieties of the maize. Okay, and this variety of the maize contains good amount of amino acids, lysines and tryptophan as compared to the normal maize. So this is our maize hybrid. Okay, which contains better amount of amino acids, then lysine, then tryptophan, then the normal maize. Okay. Now children, in the next part, we'll see some more things. Okay. Till then, you go through all these things. Thank you.